So I wanted to welcome you all. My name is Kelsey Anson. I am an educator at the DNA Learning Center in Brooklyn. So if you visited our Cold Spring Harbor lab for a camp or a field trip, we now have a new lab operating in Brooklyn, and I'm really excited to be with you today. I wanted to real quick run through a couple of things that we have going on at the Learning Center that you might be interested in, and then I will introduce our speaker for our Meet the Scientist series. But first, I wanted to share a couple of things. So at the Learning Center right now, we are actually, thought I had this pulled up. So right now we're hosting a photography competition. So All the World in New York City, this is to help build our exhibit in New York City, trying to show the diversity of the people and the culture of the city. If you live in one of the 5 boroughs, be sure to check this out. There are prizes if you are a photographer. We also have a lot of ongoing field trips. So if you're an educator, be sure to check us out for your spring semester. We can do field trips at our lab on Long Island at Cold Spring Harbor. We can do labs in Brooklyn and also at our new facility up in Regeneron in Sleepy Hollow. And then the other thing that I wanted to talk about real quick is that if you're in New York City, we have our Urban Barcode Research Project. So it is a research program for high schoolers. And right now we are taking applications to take the required courses for our research program in conservation genetics and DNA barcoding. And those are running in the evenings this spring semester and also on fall the 2 spring school breaks. So if you want more information, you can scan this QR code or go to dnabarcoding101.org. And I will stop sharing now because I am lucky to introduce you to Dr. Christopher Vakoc, who works at Cold Spring Harbor, and he's going to talk about inventing new cancer medicines.
All right, thank you, Kelsey. Can you see my slides up on the screen? I hope you can, and hopefully you can hear me okay. Yeah, I'm really thrilled to talk to you today. It's probably one of my favorite types of presentations that I like to give, is kind of just talking about my science, in the context of one's life as a scientist, and kind of going into a little bit of the origins of kind of how I got onto this kind of somewhat strange career path, and and the passion behind parts of it, some of the challenges, and and it talks about something that's very personal, which is a passion, a dream of developing new medicines that can cure cancer. And I think everyone has in their own life encounters with cancer. It's a dreadful disease, or I should say diseases. It's a lot of cancer comes in hundreds of different varieties. It mostly affects people as they age. But what has always kind of inspired me about cancer is that it can also afflict children, and that the propensity of cancer to occur in young people is what really inspires me. And I think this is what has shaped a lot of my career choices as a scientist, was to try to do something about this. And so I have my title here, which is Turning Genes On or Off with Drugs to Treat Cancer. This is kind of like our unique way to think about treating this disease. And so, yeah, I'm going to tell you today about my kind of career path, what it's like running a lab as a scientist, and try to give you, again, not just what I do now, but kind of how did I kind of get into this when I was at kind of the high school stage and beyond. And yeah, tell you about some of the struggles as well. Again, it's not always a smooth path in any kind of career path. And so I'll tell you about some of that towards the end. And so, yeah, in a nutshell, I am a professor at Cold Spring Harbor Laboratory, which is associated with the DNA Learning Center that you're familiar with. And what do I do all day? I run a research lab that is trying to develop new medicines to treat cancer. If you walked into my laboratory, which is downstairs, I'm obviously in my office, if you can see me. I am the kind of boss, or the leader, we call it principal investigator of my laboratory. But when I go downstairs into the laboratory, what you see is kind of like what's in the image here. Scientists wearing white coats, looking very serious, holding instruments that allow them to manipulate small amounts of liquids, running liquids through instruments and making measurements. This is kind of just the appearance of what it looks like in a molecular biology lab. And to be honest, when I put up an image like this, it looks dreadfully boring, and everyone's very serious, and it kind of looks the same day to day. But I can tell you, as someone that's holding those pipettes and doing the experiments, it is a remarkably imaginative and engaging experience to be the one leading these experiments and being able to understand what those manipulations are doing, and really to think about life on a very molecular level. Again, this is just kind of the outward appearance of it, but to be the one driving experiments, designing experiments, thinking about discoveries, thinking about life itself in DNA is, it's a career path that once I was one of those people in this picture, there was no going back. There was no way I was going to become a accountant or a banker. I was on this path for life because it was the most thrilling kind of professional experience I ever had, and never have had to this point in my life. So yeah, this is kind of my profession in a nutshell, and we'll unpack this. Before I do that, I wanted to just kind of give you my life story in one slide. I grew up in Allentown, Pennsylvania, which is a couple hours west of here, in a kind of suburb of this town. I was raised by a middle-class family. No scientists really in my family, no doctors in my family. My dad worked for a company doing something business related. That's as well as I understood it. I had a very sort of normal, I guess, non-sciencey upbringing. And yeah, I guess growing up, I always well in math and science, I guess, meaning that I wasn't a real hardcore student. I would never, if I look back at myself, I was not kind of an overachiever when it came to grades in school as a very young person. I was generally quite lazy and kind of doing the bare minimum to survive. And, I don't know, my parents would get mad if I, brought home grades that were below a certain level. They weren't expecting me to kind of be at the top of the class, and I really wasn't. But I would say that the classes that I tended to do better at just were math and science related. And it wasn't because I was just a really nerdy sciencey guy. It just kind of just came more naturally to me. And I see that as something I have in common with a lot of my even I see that in my own children and my brothers, that just those subjects, came more natural to us. That being said, it wasn't like an exceptional talent where everyone was like, this kid's going to grow up and be a scientist. I think I was just really bad at reading and English. I was like below average. So again, math and science just sort of, I think, to be honest, I kind of gravitated towards it because I was less bad at it. Now, of course, again, that's just me thinking about myself as a very young person. But what that naturally led to is I took more math and science classes. And I had a really formative or influential experience in 7th grade when my homeroom teacher happened to be my biology teacher. So first time I took biology. And the stories she would tell, kind of for the first time telling me what DNA was, I always remember the vignette because she was telling me how, kind of like the Jurassic Park kind of concept, that if there's a frozen woolly mammoth whose body is discovered in Antarctica or in the North Pole, if they pulled out some of the tissue, they could pull the DNA and then potentially read it into the egg from a elephant, and maybe with this DNA material you could rediscover, regenerate extinct organisms. And I don't know, there was something about that whole idea, because again, I knew what life was, but I didn't realize life had like an operating system. And once I learned that, it just sort of sparked with me, and I just sort of had this vague idea that I might be a biologist, even though I honestly had no idea what that meant as a young person. At one point, I thought I might be like a park ranger. I think I thought, again, because I was really naive. I didn't know any scientists. So like, I thought I'd like, I don't know, work at a forest or something and I didn't really know what a biologist did. Um, so again, it's with all those vague uncertainties that I kind of cruised through middle school, high school, and but ultimately found myself at Penn State. I only applied to one college. My dad sort of told me, um, we can only afford to send you to the in-state place. We're not sending you to a private school somewhere. So I went to Penn State, and I just chose biochemistry because I thought, again, the idea of DNA and how it works was kind of interesting. And to be honest, at that moment, I thought I might be a doctor, or I might be a forest, park ranger. I don't know. There's something with biology. And I went to Penn State, which was a good experience. And I think the best part about it was I started to work in a lab, because I was learning that if I wanted to be a doctor, well, you got to get some lab experience. It looks good on your resume. And so I think for all the wrong reasons, I chose to work in a lab. But once I did, there was something that really changed. And it was an inflection point in my kind of whole career. And I really just began to appreciate, again, what's shown in this slide. Like, again, it looks so boring and dry and serious, but to be the person in the lab doing experiments and understanding kind of the principles, the concepts, the technicalities of what one is doing when manipulating molecules in a laboratory, and also the culture of the lab, like being around people that are like from all over the world. Again, I'm like a simple guy growing up in Pennsylvania. I'm meeting people from China, from Africa, from like all over the world, because research is such an international enterprise. And it was just a stimulation. And like, to be honest, the friends I made in that time period are friends I have to this day. And again, it was part of that like inflection point. Once I started on this path, there was no going back. But to be honest, what I learned in that time is that science is really hard. To get experiments to work required a lot of patience and skill. And to be honest, my confidence was not very high. And I was worried that I might not be a good scientist. And so I thought, well, maybe if I can get good enough grades, I could be a doctor. And a doctor, then I don't have to worry about being a lousy researcher. And so maybe I can be a doctor and a scientist. And that's where I kind of came across this thing called an MD PhD program. And if it sounds kind of scary, it kind of is, because it's after your 4 years of college. I basically spent 8 years getting a medical degree and a PhD, because I kind of couldn't make up my mind if I wanted to be a doctor or a scientist. And in a way, I really wanted to be a scientist, but I was scared I wouldn't be good at it. And so again, from the beginning, I kind of viewed medicine as like the safer, cushier path where you'd have a certain, paycheck and like like a good life. But science was something I was like insecure about. And so I did both. And towards the end, I kind of realized that, towards the end of that 8-year investment of time, again, I'm like approaching my 30s now, still with no job, just a highly, spent a lot of time in the classroom and in the lab. I really realized that at a certain point in that process that even though I had spent a lot of time in the hospital taking care of patients, it was the lab. I had to go for it. And in a way, I think over the span of my PhD, I developed the confidence that I could actually do it. And that is probably the hardest part of a career in science, is having confidence that you can make discoveries for a living. And again, it happened at somewhere in my mid-20s. And once I had that confidence, coupled with the passion, coupled with, like not being terrible at it, I just had to go for it. Even though I could have been a doctor, like having that life, I had dropped it. And I came to Cold Spring Harbor, almost 13 years ago now, and started my own research lab, which is where I am today. And it's been 13 years. And, we have our own kind of promotion process with it. If you're an academic professor, I could tell you more about it. It's kind of boring unto itself. But I've been running a lab here for 13 years. And so, yeah, I think we all have kind of like a maybe a vision of what a professor is and isn't. Maybe I am kind of looking at me, your stereotype of a professor. Maybe not, I don't know. But what does a professor do? Well, a big part of our mission is training and education, and kind of like what the folks at the DNA Learning Center are doing. We do train high school students, undergrads, PhDs, and post-PhDs. And I would say, unlike the folks at the Learning Center, we spend probably more of our time training PhD students and post-PhD, in a smaller amount relatively of high school and undergrads. But we do, it's a mix of all those things. And yeah, it's a blend of classroom teaching, but the teaching that I'm most passionate about is research training. So I have a lab of roughly 20 people, and yeah, I'm teaching them how to do science for a living. So how to do kind of what I was doing as a undergrad and grad student, and that is a very fun thing. They learn to do science and they move on to do other things, and huge part of my job. And yeah, I make discoveries so that when those 20 people or so in my lab are doing experiments, we're trying to discover things about cancer and try to develop these medicines. I'll say more about that in a moment. We publish things, I present them, I give presentations all the time. And a big part of running a research lab, the most boring, the part I'm least passionate about is, well, you got to find funding to pay for the research and managing budgets. And, it's very businessy, but again, you got to do it because things cost money and it's got to come from somewhere. And so, yeah, I just wanted to say a few things. Again, I'm just trying to put myself in your shoes to the best that I can, but how does someone really make discoveries for a living? Again, if someone told me, like the 15-year-old version of myself, that this was going to be my future job, again, I would be terrified or confused. And it sounds weird to kind of phrase it this way, like I've done in this slide, but the reality is it really stems from a massive investment of time. Like since the time that I had that 7th grade biology class, I've been paying attention to how DNA works, how life works, how genes operate, how proteins operate inside of cells. And that knowledge is cumulative. And, again, I spent a lot of time in school, and, you get really immersed in something, just like my kids are immersed in Minecraft, and, my neighbors are interested in wine or something. You develop this passion for it, in this kind of deep immersion in it, where it feels so familiar, and like, molecule names on like your old friends. And as I've alluded to, what for me is so thrilling about science is the potential to make an impact. And we've seen this in our own research, the desire to make a curative medicine. It derives very much from passion, not from having a job where you punch a time card and get a paycheck. That is what you do have that in science, but if I can compare myself to my 7th grade classmates who didn't become biologists, well, I would say there's more of this passion embedded with what we do. People sometimes compare science to religion or art, where you're not doing it for the money. There's kind of this higher order belief that you can make the world a better place. I know it sounds cheesy, but it's an element. You can't explain a career in science by a paycheck. It's something bigger, and I think it calls out to people that want to do something in that category. Again, it's not for everybody, but I think my fellow scientists all kind of have this in common. It's kind of just this endless curiosity and love and passion for what they do. Okay, a little bit about science. Again, I really don't want to bore any of you. That's kind of my only goal of this presentation. I just don't want you to make you bored, make you think science is boring and detailed. And, again, I think back on that 7th grade biology class that I took, and, and like I said, I sat in a classroom for like most of my life, right? Learning stuff and taking classes. And I would say in my whole life, there's one thing that is the most powerful thing I've ever learned in my life, and it's depicted on this slide. The fact that we as humans are made of trillions of cells. At our origin was a single cell, whose half of its DNA came from your mom, the other half came from your dad. That DNA came together to make one single cell, which you can call a zygote. And through a process of cell multiplication and a masterful process, you can have something as complicated as each of us. Now, it gets better. because of course, DNA is the operating system that enables this sort of miracle to happen. And it's comprised of these 4 nucleotide letters, 6 billion letters long. And within this kind of very simple code is the blueprint for the, our entire existence. Me standing here giving this talk, it's my DNA, which produced my cells, which produced, et cetera, et cetera. Like, I mean, every minute of every second of our lives is kind of a function of this process playing out. And I think what really kind of resonated with me is the notion that this isn't just how I exist or you exist. It's like all forms of life operate according to this same basic code and principle. And it ranges from plants to, animals of all sorts, to microbes, like bacteria. And again, I just found this so profound. I still find it profound as I'm like kind of describing it to you. And I would say just I've never learned anything in my life that was more profound than this. And it's part of, again, that inflection point, that like never coming back once you start realizing that everything that we call our own existence is kind of a function of this kind of simple framework. Now, getting now a little bit more into my lab studies, I think what's always fascinated me, what I'm depicting here is how that one cell multiplies and produces cells that later become your heart or your blood or your liver or your stomach. Again, we're comprised of trillions of cells, and these cells are very specialized. What's interesting is every one of these cells has basically the same DNA, the same 6 billion DNA letters. DNA letters are very much kind of the hard drive of information that we're inheriting. But yet cells will turn on pieces of DNA in different ways. And so a heart cell will only turn on the parts of DNA that represent the heart genes that help a cell contract. A blood cell will only turn on parts of DNA that allow it to carry oxygen, and so on and so forth. And so I think of this as kind of the, if the DNA is the hard drive, what I'm depicting here, the turning on and off of genes, is kind of like the apps or the software that's kind of accessing that hard drive. And so, tying this now to cancer biology, this is intimately related to cancer, because what you're seeing here is a cell growing, enormously. This is sort of what cancer is. So how is cancer related to what's shown here? Is, well, every time a cell copies itself, there are mistakes made in the letters of DNA. And if that happens on the wrong genes, this will cause a cell to never stop growing. And that is essentially what cancer is. Now, the other thing I want to mention is that there are thousands of molecular machines encoded by our genes that are involved in determining whether the heart genes get turned on in the heart or the blood genes get turned on in the blood. And that is very much what my lab studies. We're interested in how genes get turned on and turned off at the right time and in the right place. And how does this process sort of go crazy in cancer cells that have acquired these genetic mutations? I'm just going to skip that. And so the one thing I want to just emphasize that is kind of a key aspect of what we do. We are kind of trying to make new medicines. And maybe something you learned in your biology courses is that the way DNA is executing its function, a lot of that function is by encoding proteins through the intermediate called RNA. DNA to RNA protein is the central dogma of biology. Now, proteins are a lot of what we are studying. And it's not protein like the nutritional substance you're used to thinking about. This is proteins as little molecular machines that are operating inside of our cells. And so we think a lot about the molecular machines that are involved in turning genes on and off in normal biology and in cancer. And if you think of a cell as kind of a assembly of different molecular machines, we're trying to find the machines that we can perturb with drugs to try to kill cancer cells selectively. And I just want to like put this one image visual out there in front of you, which is, this is what a protein might look like. It's a three-dimensional kind of blobby mess. And it often is a machine. It can be an enzyme that performs a chemical reaction. It can be a contraction protein, but it has a three-dimensional structure. And so how do medicines work? Well, they're often these small chemicals, like what's shown here is a chemical structure. And what's depicted in this diagram is a protein with a chemical sticking to it. And so how do medicines work, like aspirin or penicillin? Well, they're these little chemicals. They often stick to a protein, and they make that protein operate differently. And so this is like the design principle of medicines, is to find the molecular machine you want to control, develop a chemical that's going to stick to that machine and no other, and use it to kind of change the activity of that machine. But to do so in a beneficial way, that's not going to make a person feel worse, but make them feel better. And so we try to apply that principle to cancer research. And I just, in the last few minutes, want to just give you a vignette. One cancer that we study a lot is called acute myeloid leukemia. It's a cancer of white blood cells. If I took a drop of your blood and looked at it under a microscope with you, you'd see a lot of red blood cells shown here in this picture, kind of low magnification. And occasionally you'll see a white blood cell in here. Now, if a patient has leukemia and you take a look at their blood, what you'll see is it's massively overpopulated with these white blood cells. I know they look purple, but this is what leukemia looks like. It's this overgrowth of your white blood cells. White blood cells are supposed to like fight off infections, protect you from bacteria and so on. Well, you're making way too many of them. And these are not normal white blood cells. They can't fight bacteria. And so they're kind of crowding out your ability to make normal blood. One type of arm of research we try to do is to kind of probe all those protein machines that are inside of a cancer cell and try to find targets to make new medicines, like that green blob. A cell often has roughly 20,000 of these different molecular machines. So which among them and which kind of greasy pocket on the surface of these proteins is desirable for making a medicine, with the goal of eliminating cancer cells, but without harming your normal cells. And so we have sophisticated technologies, some of which we invented, to ask this question, to take thousands and thousands of different protein molecules. And so like this is an example in this slide I'm showing. There's all these different molecular machines. They all have their own 3D structure. They're all very complicated. They have cryptic names, profoundly complicated to study them. But which among them, if you could make a medicine like penicillin for one of these targets, which one do you go after if you want to cure a child of leukemia? So that type of question is what fuels research in my lab. And one of our favorite discoveries goes several years back now. We found one of these targets. And in fact, the green blob I was showing you is a target we discovered for leukemia. And that's an actual drug bound, a medicine bound to that target. And then the punchline of this is we discovered the leukemia cell is kind of addicted to this molecular protein machine. And we could show that if we designed a medicine that sticks to that protein, it changes its function, which is involved in turning genes on and off. Well, we could make these mice with leukemia. So we could take leukemia cells from a human, allow them to grow uncontrollably in animals, and now treat them with these medicines that hit this target we discovered. And we showed that these animals will live longer if we treat them with this drug. And so that was kind of the first step towards trying to make a new medicine. Within a few years, several drug companies took this discovery into the clinic. And several years later, we started to see the first patients with leukemia being treated with this drug that we discovered. And so this is some clinical data from a 74-year-old woman. So this is an elderly patient with leukemia treated with a BRD4 inhibitor. And this black line, what it's measuring is the amount of leukemia cells in her bone marrow over time being treated with this drug. And you can see the black lines going down. What's also exciting is the dark blue line here is her normal white blood cells. Again, the ones that fight off bacterial infection. And what you see is after maybe 50 days or so taking this drug, her normal white blood cells started to come back. And so this, again, I forget about all the details. All I'm trying to emphasize here is like from my research lab here at Cold Spring Harbor, we are doing research, studying molecules and trying to find kind of the Achilles heel or the weakness of a cancer cell, trying to find a molecular machine that we can manipulate with a medicine so that we can help patients with cancer recover from their disease without having to be tortured by chemotherapy or surgery or radiation therapy, which are often the therapies that are used today to treat cancer patients. And as you know, these are devastating therapies on top of devastating diseases. What we aspire to is to develop medicines that will be not only effective in melting away tumor, the black line here, but safe, like the blue line here, allowing normal cells to kind of come back and take over and kind of resume normal function. And we dream of a day when patients won't have to take chemotherapy, but that they can take this new generation of safe medicines. And we hope to kind of guide that process through our discovery research. And so finally, I was asked to kind of mention obstacles. So I'm trying to express hope, and it's fun for me to kind of talk through kind of highlights and thoughts, inflection points in my own career. But it's not always so easy to navigate a career in science. I already talked about confidence being a big challenge. I think everyone respects science. I think most people respect science, right? Finding solutions to disease, people don't die of smallpox anymore, or measles, or there are diseases that humanity through science has solved. And you can get on a plane and fly around the world and land where you intend to go with minute time scale resolution. We have fun. We all respect and appreciate technology and science, and especially now more than ever, I think, living through a pandemic. But I think the hardest thing is having confidence in oneself that you can actually do it and make discoveries. Again, I did not have that confidence for most of, certainly when I was your age. And I can tell you, like, I would say I had unusually low confidence as a teenager, as a child. And I'd say one reflection of that is I never once raised my hand in the classroom in my entire high school, elementary school, or college education. And it wasn't because I was trying to be cool or something. I literally just never had the confidence to ask a question. To me, answering a question was frightening, for fear of saying something dumb or sounding foolish or something you should have known, or I don't know. I just never had a lot of swag or a lot of confidence to do that. And for me, I would say I was pathologically shy as a young person. And it's weird for me to say that, because nowadays, I basically speak for a living, right? I present science. I'm talking to you. I might not look shy or introverted, in this Zoom presentation. But I can tell you that was very much something I learned in my 20s. And I think it was because, in some ways, I think it's one of the most enduring parts of going to medical school, is that you were forced to give presentations all the time, every day. And and it was anguish for me. You imagine this shy kid giving presentations every day to like doctors, senior doctors in the hospital. And it kind of beat out of me some of these shyness. Like, it kind of forced me to have to learn how to be comfortable. And so again, I see that as not unique to me, but I think in some ways science looks so profoundly technical. It can get so detail oriented so quickly the moment a scientist starts talking about what they do. But it can be challenging to kind of find confidence when there seems like it's so impenetrable as a career path. But again, I think it's important. Again, so that's the obstacle, and kind of how did I overcome such obstacles? I said part of it was like I was forced to give talks all the time, and that helped. I also had very generous mentors, I think, who took the time to talk science with me one-on-one, and they never made me feel embarrassed to ask questions or be naive. And so I know it's hard to find mentors. I think mentors are always in short supply, the great ones, but I was very fortunate to find them. But again, most of the people I was surrounded around, like in college, medical school, etc., I would say we're not those kinds of mentors. So I think it's kind of trying to find those types of people that was really influential, and kind of seeking them out whenever I could was key to kind of developing confidence. So that's my last slide. I hope you weren't bored. And if anyone has a question, I'd be really thrilled to answer any questions you may have. So thanks again for the invitation, and yeah, I hope you learned something. Thank you.